Hi, I'm Karol Prieto. I am postdoctoral fellow from Dr. Kenneth Tsai Lab, and today I am presenting our work denominated Pharmacologically Induced Oncogene-Driven Senescence to Sensitize RAS Mutant Cancer to Immunotherapy. This work is based in the point of most of 30% of the human cancer are driven by RAS mutations. One of the main things that we know right now is the melanoma patients that have a BRAF mutation. They are responding to the therapy through the mechanisms or activation of ERK pathways. And then when we have a patient with a BRAF mutation, the patient responds decreasing ERK signaling. However, when some of the patients that are resistant to the therapy, they have an increase in this signaling of ERK based in new NRAS mutations. And then with this in mind, that we have two pathways that are working together, we start to think about the problem of the regulation of ERK pathway activity. Then we know that the inhibition of the ERK pathway will decrease the proliferation of some cell lines. If we have a intermediate point, we will have an activation in the proliferation, but if we have a hyperactivation of the ERK pathway, probably we will have a decrease in the proliferation. And then to make this point, we use a different NRAS melanoma cells that have a high activity of ERK. The ERK pathway. And then when we treat the cells with BRAF inhibitors, dabrafenib, encorafenib, and bemurafenib, we saw a decrease in the proliferation in most of the cell lines. However, when we use PLX 8394, that works without the regulation of the ERK pathway, we didn't see the effect. And then following that, we try to see what is happening in the ERK regulation, and we, of course, observed an increase in the phosphorylation of ERK pathway. In addition, we saw that this is related with an increase in the senescence of the cell lines, and then we conclude that the low concentration of BRAF inhibitor decrease the proliferation of NRAS mutated melanoma cells, and also this is based in the senescence of the cells. Then following this, we did an in vivo experiment to see what is happening there. And then when we did the combination of dabrafenib in a NRAS model, melanoma model in vivo, what we saw was compared with the control, we have anti-PD-1 response, dabrafenib response, but the combination of dabrafenib and anti-PD-1 is the only combination that make a better response and to the long time. This is mainly based in we saw that this animal have an increase in the cytokines. In CD8 T cells, we saw an increase in interferon gamma, TNF alpha, and CD107. But in addition, we saw a different cytokines in the three models of cell line that we evaluate. We evaluate 501 male, IPC 298, and male U. So in these cell lines with the treatment of dabrafenib, we saw an increase in all of these cytokines. Moving on, is not only NRAS mutated cells are responding to BRAF inhibitor. We did the experiment also in KRAS cells in a pancreatic model, and we saw that the all cell lines with KRAS mutations have the same response. We use low doses of dabrafenib and encorafenib to see the same response, and we saw the same in the KRAS model. Then we have melanoma cells, NRAS mutated, pancreatic cells, KRAS mutated, that are responding to BRAF inhibitors. And then with this in mind, we demonstrate that here that the preclinical validation of a strategy sensitizing the RAS mutated cancer through the use of BRAF inhibitors can increase the ER pathway, and it will be related with more senescence and with a decrease in the tumor in the patient and also in the animals.